Good morning, children. In last class, we learnt about some cell organelles. Those are endoplasmic reticulum, and we also learned about ribosomes, and we learnt, that is endoplasmic reticulum. Mitochondria we learnt. So today we are going to see some other cell organelles which float in cytoplasm, and their functions we'll discuss today, children. So our today's topic is cell organelles. In cell organelles, today we are going to discuss about Golgi bodies. Where Golgi bodies are present? Golgi bodies are also present in cytoplasm. In cytoplasm, they are floating. How ribosomes float in the cytoplasm, how mitochondria is present in the same way, Golgi bodies are also present in cytoplasm. Now we will see the structure of Golgi bodies. They are membrane bound vesicles. Membrane bound means they are attached to the membrane. Cell membrane. Which membrane is present in the cells? Cell membrane is present. So they are membrane bound organelles. Means they are attached to the membrane. Cell membrane attachment is there with Golgi bodies. Attachment. They are having attachment with cell membrane. Membrane bounded vesicles. So they are parallel to each other. One side they will have connection with cell membrane and the parts are parallel to each other. The scientist Golgi found these Golgi bodies first time in cytoplasm, and for his invention he also awarded by Nobel Prize, and he also said the functions of Golgi bodies. Now we'll see the functions of Golgi bodies. What functions it performs in each cell. It collects the materials from ER. So it's having connection with the nucleus and the cell membrane. In between it's floating. So it collects the materials from ER. Actually, the smooth ER, yesterday we discussed in our video, the smooth ER is synthesizing lipids and proteins. So this Golgi bodies collect that lipids and proteins and take them and give it to the cell membrane. And these lipids and proteins are helpful for the formation of cell membrane. So it's collecting the materials from ER and passing to the cell membrane. So in this cell membrane, preparation of cell membrane, those lipids and proteins are used. That means Golgi bodies are storing sometime the materials which are prepared by the other cell organelles, and they are also dispatching where actually they are needed. Sometimes they are storing and later they are dispatching where exactly this cell organelle or these nutrients or the synthesized nutrients is useful to that particular place. They are dispatching. So those are the functions of ER. Those are the functions of Golgi bodies. Along with that, they are also helping for the formation of lysosomes. They are helping for the formation of lysosomes. About lysosomes we are going to discuss now. So for the formation of lysosomes also Golgi bodies are useful, and they are also preparing complex sugars. Complex sugars are prepared from simple sugars. So your carbohydrates are later converted into like sugars that you learnt in your digestion lesson. So these small sugars all will combine and make a complex sugar with the help of Golgi bodies. Same way they are also helping for the formation of lysosomes. They are collecting materials from one cell organelle and dispatching the place where they are needed. So those are the functions of Golgi bodies and found by Golgi, a famous scientist. Okay. Now we will see what are lysosomes means. What actual function they'll do in a cell. Lysosomes are single membrane vesicles. They are also membrane bounded vesicles connecting with the cell membrane, and single membrane is present. Inside this lysosome, powerful digestive enzymes are present. So this is the structure of lysosomes. These lysosomes have a single membrane, and inside this lysosome, powerful digestive enzymes are there. Digestive enzymes are present. So these enzymes are helping to protect the cell from infections. Any infection, how we'll get the infection actually? If any particle enters into our body, the foreign particle like small germs like bacteria, virus or any other microorganism, when it enters into the body, with the help of these powerful digestive juices, it can engulf, it can deactivate that microorganism and send out from the body. So it's helping for the digestion of foreign particle. That means it's helping intercellular digestion. So that's why lysosomes are also called digestive sacs. We are also calling them as digestive sacs. Why we are calling as digestive sacs? Because they are digesting the foreign particles and helping us to protect us from infections. And next, it can also, these digestive enzymes are also helping to digest the worn out parts. The cell parts which are not working, those parts are get digested by the help of this digestive enzymes which are present inside the lysosomes. So they are worn out cells are destroyed cell parts. If any cell organelle is not working in that particular cytoplasm or in that particular cell, those can get digested by the digestive enzymes which are present inside the lysosomes. So that function the lysosomes are carrying out in a cell, and they also destroy themselves. They can get destroy themselves when the particular cell is not working. It get destroyed by the juices, by the digestive enzymes present inside the lysosomes. So it is destructing itself. That's why they are also known as, lysosomes are also known as suicidal bags. We can also call them as suicidal bags. Why we are calling them as suicidal bags? Because they are suiciding themselves to give out the energy. It destroying that cell and giving the energy. Because of this reason, when we are doing any fasting, any festival time, also when we are in fast, also we get the energy because of this lysosomes. And any worn out cells, any damaged parts were there, they get digested by the lysosome enzymes, digestive enzymes present in lysosomes. So those are the functions of lysosomes. Till now we discussed about Golgi bodies and lysosomes. So this formation of lysosomes, the Golgi bodies are helping. Now we will move on to the next cell organelle. Next cell organelle is, so Golgi bodies are also present. Golgi bodies are also present in plant cell and animal cell. But the Golgi bodies in plant cell are known as dictyosomes. Dictyosomes. Golgi bodies present in plant cell, Golgi bodies present in animal cell. But in plant cell we are calling them as dictyosomes. The function is same. What functions the Golgi bodies are carrying out in animal cell? The same functions dictyosomes carries in plant cell. So now we will move on to the third cell organelle, that is vacuole. Vacuole is also present in animal cell and the plant cell. But only the difference is in plant cell the big vacuole is present. Because of this big vacuole, the nucleus can be pushed to a part. For example, we'll see a plant cell. This is cell wall, only present in plant cell. And because of this big vacuole, the nucleus can be pushed to a side. And this is the vacuole. It is also a single membrane like our lysosomes. Just now we discussed lysosomes are single membrane. Same way the vacuole also single membrane, and it is covered by a membrane known as tonoplast. What we call it? Tonoplast. The membrane which is surrounding the vacuole is known as tonoplast, and the liquid present inside this tonoplast or inside the vacuole is known as cell sap. Cell sap is present inside the tonoplast. Inside the vacuole, cell sap is present. So this vacuole is surrounded by tonoplast. And this cell sap is helpful for storing amino acids. From where we get the amino acids? From the digestion of proteins, we are getting amino acids. This by the synthesis of proteins, we get the amino acids. So this cell sap is helping for the storage of amino acids. It can also store sugars. And it can also store some organic acids. So for the storage of this, for the storage of amino acids, sugar and organic acids can get stored by the help of cell sap. Where this cell sap is present? Inside the vacuole. That means the function of the vacuole is storing. It can store the proteins, amino acids, sugars and organic acids. These all are stored in vacuole. And it can also store water. Along with this all, the vacuole is also helping to store the water. Why the vacuole is bigger in size in plant cell when we compare with the animal cell? In animal cell, the vacuole size is small, whereas in plant cell, the vacuole is very big. The only reason is it needs to store the water. For storage purpose of water, a big vacuole is present in plant cells. So understood now? One is Golgi bodies we discussed. Next lysosomes we discussed. And then vacuole we discussed. Now we'll see one more cell organelle, which is very important only in the plant cells. That is, this is present only in our plant cell. These are plastids. The next cell organelle is plastids. Plastids are present only in the plant cells. They are absent in animal cells. And there are three types of plastids present in plants. In plant cells, how many types of plastids present? One is chloroplast. Second one is chromoplast. And the third one is leukoplast. So three types of plastids present only in plant cell. Now we will see the structure of these plastids. how the plastids are and what purpose they are helping, we are going to discuss now. First, we'll see the first plastid, that is chloroplast. Chloroplast is oval in shape and double layered membrane, oval in shape. This is outer membrane, and this one is inner membrane, and the shape is oval shape. What structure we are discussing now? This is chloroplast. Chloroplast structure we are discussing. Where this chloroplast actually useful? The very essential to the life on planet Earth is possible because of this chloroplast. It's present only in plant cells. Because of this, the plant parts are getting green color, like their leaves, their branches, sometimes their stem appears green because of chloroplast present in them. So about that structure we are discussing. So with the help of chloroplast, which function takes place in plants? The very important function, photosynthesis. Which function takes place with the help of chloroplast? Photosynthesis is taking place with the help of chloroplast. So the structure of chloroplast, the shape of the chloroplast is like oval in shape and it is double membraned in structure. Inside this inner membrane, a liquid, how our cytoplasm appears in structure like jelly-like structure, same way in chloroplast, inside after the inner membrane, the jelly-like substance is present, liquid substance, that's known as stroma. So what we call it is stroma. So inside this chloroplast, stroma is present, and in the stroma some thylakoids, grana thylakoids we call. The structure appears like this. If we arrange coins one above the other, how this structure looks, how this structure appears, how this structure is looking when we arrange the coins one above the other. Same way, inside the chloroplast, along with stroma, the thylakoids are present. These are known as grana thylakoids. They are known as grana thylakoids. One granal thylakoids are attached to the other grana thylakoids by stroma thylakoids. These attachments are known as stroma thylakoids. So this is the structure of chloroplast. Oval in shape, double membrane, inner membrane after a jelly-like substance called stroma present. Inside the stroma, granule thylakoids are present, and two granule thylakoids are jointed by stroma thylakoids. Finally, the whole chloroplast structure is helping to convert the sunlight into light energy into chemical energy. That you learnt in photosynthesis, and the reactions, two types of reactions takes place here, light reactions and dark reactions, that you will read in class 10. Now you understand that the chloroplast structure is helping to convert The chloroplast is helping to convert, chloroplast structure is helping to convert light energy into chemical energy. It's converting light energy into chemical energy. Who is converting like that? Our chloroplast is helping to convert light energy into chemical energy. They'll store the sunlight's energy in the form of photons. They'll take the energy in the form of photons and they will convert this into like glucose plus oxygen. Who is helping here? Chloroplast is helping. So it's converting that light energy with the help of water molecules. With the help of water molecules, the chloroplast is converting light energy into glucose and oxygen. And whereas oxygen is a byproduct in photosynthesis, which is not useful for the plants, so that they are releasing outside. That oxygen we are using to carry out our respiration process. This is all known story for you. The new story is here, how the chloroplast is helping to carry out the photosynthesis process you learned now. So this way the plastids are present in the plant cells and helping them to carry the basic life process of photosynthesis. And then next type of plastids when we see, those are chromoplastids. Those are chromoplastids. These chromoplastids help to give the color to flowers and fruits. Flowers and fruits we are getting in different colors. Different flowers are available and different colors of fruits we are getting because of these plastids. If chromoplastids present in them, they appears in different colors other than the green color. Green color appears because of chloroplast. Like your pomegranate, when you see, the seeds are in which color in pomegranate? Red in color. That red color comes because of chromoplastids present in them. In same way, flowers, n number of purple flowers, roses are available in different colors. Those all are possible because of the presence of chromoplastids. And the last one is leukoplastids. Leukoplastids gives white color to the plant parts. They are giving white color to the plant parts. Like some flowers like jasmine available in white color. They are getting white color because of the presence of leukoplast. They gives white color, no color, white color to the plant parts. Leukoplasts are giving white color to the plant parts. So till now, how many cell organelles we discussed? We discussed about 4 cell organelles. One is Golgi bodies and the second one is lysosomes. Third one is plastids and the next one is vacuole. So understood, children? So Golgi bodies present in plant cell and animal cell. In plant cell we are calling them as dictyosome and vacuole present in plant cell and animal cell. Only difference is in animal cell vacuoles are small in size and vacuole is surrounded by a membrane known as tonoplast, and inside the vacuole we are having cell sap that helps to store, and lysosomes are acting like suicidal bags because they have powerful digestive enzymes and they can digest and they can destroy the worn out parts in cell. So we are calling them as suicidal bags. And then plastids are present only in the plant cells and their structure appears like oval in shape and they are helping in photosynthesis process. Understood, children? So up to here, the major part of cell organelles we have completed. And in every cell, the centrally located which part is present? Yes, that's the nucleus. So about the nucleus, we'll discuss in our coming class what are the functions of nucleus, structure of nucleus, everything we'll discuss. And your today's homework is draw the diagrams of Golgi bodies. Draw the diagrams of Golgi bodies, lysosomes, vacuole, and plastids. Draw the diagrams of cell organelles like Golgi bodies, lysosomes, vacuole, and plastids. What all we discussed in our today's class. Draw those diagrams in your notebook neatly and label them. And many of you are posting your homeworks through WhatsApp. Well done. Very well. Your diagrams are so neat and labeling everything is so perfect, children. And the others who are not yet started still, you please complete all this and post it to me. Thank you.